Much for making me sound smarter than I am. Thanks for the introduction. Thanks for having me here, and thank you to you three for helping us. I really appreciate it. I felt welcomed since the get-go. One thing that I also really appreciate is that you are giving young researchers the possibility to present. I like that. That is something that everybody needs to sort of learn if you're in science, and you gave really nice flash presentations yesterday. And so let's see if I can sort of like keep my end to this. So this one works, this one does not work, or is the other one? More or less. So yeah, I've been working with Vincent Ising. I'll show a picture a little bit later on for a long time, and also with Gustav. We started collaborating back in 2003, working on enzymes using water to break, like I say, bonds. And then we had this discovery back in 2010, which I'm going to talk about. So we have funding from ERC, the ERC Synergy grant. Also, we have some funding from Oxytrain, which is ITN network previously. And now I'm not going to talk about that right now because it's basically a secret, but I also have a project with Tomas and Arthur, and I really appreciate their expertise. And as you can see, there's a lot of people who's been working, and I'm going to show most of their faces as I go through it. So one little critique that I have is that my training is original organic chemistry. You turn like organometallic and inorganic chemist, and then turn into a physical chemist, then turn into a biochemist, then turn into a biotechnologist. But I'm still a chemist at heart. And I really like to see the pictures of the enzymes. Because I mean, a lot of these cool enzymes, I got this paper from Agnieszka this morning, or she sent it, and it's something I've never seen before. It's pretty cool. I like to see that. So that is one slight critique I have. Show the enzymes. I know it's biotransformation, but it's basically all chemistry. And here we have this copper center. It's ligated by two histidines. It's really unique because one of them is the N terminal, and then it also provides the back tail to bind. And that is quite unique in nature. And there's some other stuff as well. I'm going to show that a little bit later on. They're small, 200, 250 amino acids, about 18 kilodaltons. Every one of these have the exact same structure at the active site. It's a complex second sphere, as we call it. I'll come back to a little bit later on. And this binds to either cellulose or chitin, like this really nice insoluble biopolymers. So to do that, it's really nice and flat. There's some variations, and you also may hear like this C-terminal domain we call like carbon binding modules. So this is a history of LPMOs. It's not something I put up. It's in this review paper, and it's almost correct. The main difference is that the name Lytic polysaccharide monooxygenases, which I guess is wrong. We proposed it in Achman et al. PNS 2012, and not in this review that they point out. But basically, it starts in 1950s with Elvin Rees, and there's a lot of different discoveries through the time, and I tried to pick some of the most important one. And this is pretty cool. Elvin Rees, back in 1950, really nice Canadian microbiologist. Also, since he was good, he got his name, Trichoderma reesii, very famous fungi that we use for production of enzymes and everything. It comes from him. So he basically said, okay, we're having this really tough substrate. And the enzymes that we thought were degrading this, or we know degrade this, glycoside hydrolases, they cannot do them by themselves. It's just too tough. So it has to be something else. And he posted a C1CX hypothesis. And like he states in his paper, he doesn't really know what it is. And he points to other researchers previously on. You had this paper by Arantes and Sadler, who thought that, okay, you're loosening the change, but they did not find what it is. Back in 74, a Swedish group, they could see that, okay, oxygen is important if you break down cellulose, and only valid when cellulose alone is present, suggesting an oxidative enzyme, completely away from the classical where it's just use of water. And again, Elvin Rees, he looked at 100 different bacteria degrading chitin. We work a lot of chitin, the one you find in shrimps, crabs, which is a really huge advantage, actually, by the way, because, I mean, there's not too many people there, and then, you know, we can get like this competitive advantage. Another thing is that Elvin Rees said that, okay, Serratia marcescens has the perfect chitinolytic machinery. And this study back in 98 by this Japanese group, they basically placed its Serratia marcescens on chitin and discovered 1 glycosidase, 2 glycosidase, 3 glycosidase, 1 biase that cleaves dimers into monomers, and then 1 they call chitin binding protein 21. So basically it's 5. So that's 2 going from each side, 1 on the top, 1 producing dimers into food for bacteria, and there's 1 more. So what is it? So Gustav, back then a postdoc, he's a professor now, he produced this one, made a crystal structure. Again, you can see it's really nice flat surface. If you put it with a chitinase, mine 500 hours, you get slightly increased action. Simultaneously, Mats Sangren at Umeå, he solved this structure of a another enzyme. It looks fairly much the same, but this one is active on cellulose. This is active on chitin, flat binding surface. Here like a nickel in the center, so it didn't show the activity, but it basically showed that there's a metal binding site. And then Gustav, him and I and Vincent, so this is my colleague Vincent, Gustav and Bjørge did main bulk of the work. So these are the hands and the head. This is the head, this is the head, and then I just, you know, tag along. Gustav, inspired by this work, went to this conference in Venice, and he saw these papers saying that, okay, there's a metal center, let's try different types of metals. So we tried all different types of metals, and he was really annoyed that iron would not stay in iron 2. It just popped out to iron 3, which it normally does. So then they put ascorbic acid to keep it reduced, and he saw this like gigantic explosion of activity. And if you ever apply for a job with Gustav, it's always going to ask what's the most important experiment. It's a control experiment. So we basically did the experiment without iron just to check that the background what, and then we saw it was still active. So that's pretty cool. Another thing I'm fairly proud of, this is all done by NMBU, Norwegian most smallest university. So we took a little bit swagger for that. So basically, as you can see, this is hours instead of weeks. So we found something. So what is it? Well, this is my contribution. I thought, okay, if it's oxidative work, then let's just label oxygen with O18. And then we actually saw an increased mass of 2. Let's label the water with O18, and we also increase the mass of 2. So we proposed this mechanism. Oxygen, 2 electrons, 2 protons, and water going in, and we get this nice acid instead of like a new sugar. We also see that if you do cyanide, it emits, and if you do under anaerobic conditions, nothing happens. So it's an oxidative mechanism. So we wrote this up, put this in Science, the highlight of my career, and then one of the co-workers of Elvin Rees sent Vincent this email: "See, one lives, Elvin Rees is turning in his grave." I thought it was quite nice. So we saw that this one can work for chitin. This other insoluble polymer is also cellulose. They're very similar. I should put the structures up. I forgot that. I'm sorry for that. But they also work. We sent this to Science as well. They didn't want it. That's fine. So we sent it to Protein Science on a Sunday evening. Monday morning it was accepted. It's really cool. It's really nice sightly. And a lot of credits to Sara Forsberg, which is the queen of this type of LPMO. She's been with us for a long time. So we also see synergy with the cellulases, not as much as it did for CBP1, but that's a completely different story. But we do see oxidized oligomers when we incubate with this type of LPMO. They also work on hemicellulose. It also works on starch. We suggested the name Lytic polysaccharide monooxygenases, suggestion by Gideon Davis at University of York. He's a guru in the glycoside hydrolysis field. And currently there are 8 different families. Bernard Andressa at University of Marseille, or the right now at I think DTU in Denmark. Him and Pedro Martinez, they have classified all carbohydrate active enzymes into different family based upon their sequence, and it's really nice. It's really helpful. And then he has put 8 different families for our LPMOs. So I said a little bit of history. It's not just us. We missed the medal. And this is basically Gustav and my fault, because I mean, when we did this, Quinlan et al., Paul Walton and Gideon Davis, they tried copper. It works with copper. They can see it is copper in the crystal structure. They can see about EPR. We also tried with copper, but then Gustav, we like a huge overload of copper. And basically what happens then is that it scrubs away all oxygen, you take away all the hydrogen peroxide, and you basically sit there with nothing to react, and you need this. I told this to Gustav twice. I should do it 3 times, but that's fine. The cool thing is that we got someone from York who are more like inorganic chemists to also be interested in this, because we need to know someone who also understands physical chemistry and inorganic chemistry to push this field further. That's a good thing. So that's the first EPR study. Paul dubbed this the history embrace, because you embrace. I thought that was quite nice. And again, you have this N-terminal coordination to the copper too, which we think is really important for its properties. And they also show that it's oxidative. And then we have this brilliant postdoc, Bastien Bizarro from Toulouse. He's back there now as an independent researcher. He was inspired by a work by Canella in Copenhagen, thinking that what they observed, they saw that this chlorophyllin, if you added that to the reaction, maybe I should have shown that as well, you get this elevated reaction rate. But he thought that, okay, what they saw was basically just production of hydrogen peroxide. So what did he do? It's pretty cool. He borrowed my thought, maybe labeling with O18, and as little as 25 micromolar hydrogen peroxide in the presence of up to 1 millimolar. Normally it's like 200 micromolar, but you can also saturate. you get like a huge change in rate. Sorry. So he thought that, okay, it is not a monooxygenase, it's a peroxygenase, it's much faster and more efficient, and you can also do this in the absence of O2. So you can take away all the oxygen, just hydrogen peroxide, it still goes. So it's a peroxygenase. This is very convincing. And then you can also use sub-stoichiometric amounts of ascorbic acid. So this is the reaction. You start with copper 2, you put electron, and the cool thing about LPMOs, and that is also one of the reasons I like that you guys show what type of enzymes you have and what types of electron donors, because for like most iron proteins, you need certain specific electron donors. For this one, you can put whatever. Doesn't really matter. Gallic acid, cysteine, phenolic compounds, you can use monolignals, you can use ascorbic acid, you can put whatever. It's really, 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 really nice. So then you get into copper 1, and then typically you put hydrogen peroxide, you get cleavage, you can turn over and over and over and over and over. You just need this priming reduction. You can also make like heme proteins use hydrogen peroxide, you shunt. But then you start with iron 3, you make compound 1. I don't understand why it is more unstable if you shunt compared to if you build up compound 1 using oxygen. I don't understand. But they basically lack this general acid base to stabilize compound 1. And it's slow. 1 per second, 0.1 per second. This one here is up to 100 per second. Another cool thing and also very central thing is that if you don't have your carbohydrate substrate and you can put just oxygen and you can reduce to superoxide and then you reduce to hydrogen peroxide and you have this oxidase mechanism, which is really important. We use this to sort of like characterize our LPMOs and place them in categories what they are. I'm going to show a little bit more about that. And also, it is also important to understand why we now understand what we thought we understood back then. I'm going to try to explain. The bad thing is, and that is something that I think we should stress a bit more, is that if you don't have substrate, you may inactivate. I'm going to show where this takes place. But it can also happen, I was going to show that, when you have the substrate. Because this is really, really powerful chemistry. I mean, you have the power to break bonds of like 100 kcals per mole. So it takes everything. So if you do have uncontrolled conditions, not uncontrolled, but if you don't have everything correctly, then you create this OH radical, and this OH radical starts chewing up your amino acids close to the active site. Again, this is a work of Bastien Bizarro, a brilliant postdoc. So we thought this was a monooxygenase. It's not, it's a peroxygenase. So how come we thought it was a monooxygenase? Because I mean, I showed experiments where we label oxygen, but what happens is that if you don't provide hydrogen peroxide, the system provides hydrogen peroxide, either by this oxidase activity of the LPMOs. You can also have this reductant auto-oxidation. Ascorbate produces hydrogen peroxide, cysteine produces hydrogen peroxide, gallic acid produces hydrogen peroxide. So that's also why I talked to you yesterday about like checking your system with what other systems are for producing hydrogen peroxide. So the rate depends on the LPMO. Bacterial, they typically produce low amount. Fungal produce high amount. The reductant, the LPMO reductant interactions, the substrate, the presence of transition metals, the pH, everything counts in. And if you'll go to the literature, so Anton Stepnov is one of the most brilliant PhD students I ever met. He went critically in all different types of works across basically the world, using cellulose 2, which were our second LPMO, And they looked, even including our studies, that you can get kcat from 0.2 to 7.9 per minute, depending upon the conditions. So he spent 3 years, made Vincent, my colleague, extremely frustrated, but now he produced this paper, which is really, really highly cited. It's closing to 100 as well, and he saw that, okay, if you don't add any hydrogen peroxide, use gallic acid, you can break down a certain amount in 20 hours. If you use ascorbic acid, you can break a certain amount down in 24 hours. If you take hydrogen peroxide, you break the same amount in 60 minutes. And also show that the production of H2O2 is pretty much independent of the LPMO. So if you take ascorbic acid in the presence of the LPMO, you get this much amount of what did I do? There you go. So if you take this amount of LPMO, 1 micromolar in the presence of ascorbic acid, you produce this hydrogen peroxide. If you did free copper, a lot more. Also, this is one of my most favorite figures. A little bit tricky. It's not tricky, but yeah. Anyway, if you break down, in this case, chitin, and you only add either ascorbic acid and gallic acid, which then in turn produces hydrogen peroxide. You take just a little bit of LPMO, you get the reaction going. But then if you increase the amount of LPMO, and you know, typical Michaelis-Menten, that it's a proportionality between enzyme concentration and enzyme rate. And you don't see that. It's independent of the LPMO. So what happens here is that it starts to bind to the substrate. And if it binds to the substrate, it doesn't produce hydrogen peroxide, it goes slow. Another brilliant PhD student, Lucas Reeder, is now a postdoc in the project with Tomasz, Arthur, and me. He cloned our LPMOs into P. Apostores. That's why I also asked about the expression levels you get, the scalability, what kind of systems you use. I mean, there's a lot of different types of systems we can use, and I understand that you need to use E. coli. But he is basically like a wizard on producing proteins, and he produces a really cool one called AFAA11B. So AF is Aspergillus fumigatus. AA is auxiliary activity based on Henri Bassard's classification. 11 is the family it is, and B is the second one we did discover in the gene. The cool thing about this one is active on soluble, because I mean normally these are active just on insoluble, either cellulose or chitin, but this one's actually active on soluble, and then we take that into our advantage. And I'm not going to go too much into the detail, but this one here has a redox potential. Let me see, where is it? Here. Yeah, here, which is much lower than free copper. And then it's also better at giving electrons to oxygen. Then it's also better to bring electrons to produce hydrogen peroxide. So it's a huge oxidase activity. So if you compare with the first one that we discovered, high potential, real crap at producing hydrogen peroxide. So we used as a measure to sort of characterize our LPMOs. Back then, we were still trying to prove to the world that these are peroxygenases and not monooxygenases. And what can you do to do that is that if you take horseradish peroxidase, which eats hydrogen peroxide, you observe that you don't get any reaction. And it's sort of like the more you put, the slower it gets. So if you scavenge hydrogen peroxide, then you don't have any what we call like the monooxygenase reaction in the presence of this this soluble substrate. And this is basically like what the Anton saw. It's the rate is LPMO independent. We took one step further. We had this other one, Neurospora crassa A9C, also very cool, also active on soluble. This on cellulose oligomers, and this is a collaboration between Anton and Lucas. And to make a very long story short, we saw the same thing. So if you have a horseradish peroxidase, it does not work. If you don't, it works because, you know, the LPMO produces H2O2. We got this critique from Paul Walton at University of York, very nice guy, very stubborn guy. I visited him for a month back in 2018. He said that this is a monooxygenase, and then that the peroxygenase is just like this crazy side reaction. But then Lucas showed that, okay, you can actually get rates up to 100 per second, and we only observe specific oxidation. And the cool thing about NMBU is that we have a really nice instrumental park for analysis and everything, and we can analyze whatever we want. And here we can see that either if you use just oxygen and then H2O2 is produced, or if you use hydrogen peroxide directly, we get the exact same products. Very convincing, in my opinion. So it's specific. And we're not the only one. This is a really cool experiment. It's not us, it's Roland Ludwig at Boku. Very smart guy, very nice guy, very friendly guy. And he had this really nice experiment I really want to show, is that he has this cool sensor that measures concentration of O2 and H2O2 in situ with time, high resolution. And then he sees that the O2 concentration goes down, the H2O2 concentration goes up. And then he has this cellulose, I'm sorry, you see the H, one giving electrons and one producing H2O. And when he adds the LPMO, H2O2 goes down, oxygen stays the same. It's a peroxygenase. And even though Paul was very skeptical, this is another work also with Roland Ludwig in collaboration with Vincent, my colleague. He's using the same sensor, and it titrates in hydrogen peroxide time after time after time after time after time after time after time. And you get turnover numbers more than 30,000. This is a peroxidase. So I'm doing today what I did yesterday. I talked too much, but half an hour. Half an hour? Cool. Good. So this is the key. The copper N terminal, really important, because I mean, we believe that having this non-pi donating ligand is one of the reasons why we have this activity for the copper. So this is the histidine brace, but LPMOs are much more besides the histidine brace. And the first thing is what we call second sphere residues. So the histidine brace is universal converse. It always looks like that for all LPMOs across nature. But then we have other residues. And I'll go into detail about those and also their importance, because we believe that those are the ones that make an LPMO an LPMO. The copper histidine brace is copper histidine brace, but then how we tune this copper, you tune it by the second sphere residues. So, firstly, all LPMOs have either a glutamine or a glutamate. So to see where they are here. They're all placed in the same place. Exception is the bacterial ones where the glutamate is here. Fungal typical have glutamine for the ones active on cellulose, and it has glutamate for those active on chitin. And if you have a glutamine, you're accompanied by another histidine and a tyrosine pointing from below in protein. If you have a glutamate, you have an asparagine and a tyrosine pointing from below. If you're bacterial active on cellulose, you either have the same as the fungal ones, or you can have an glutamate, asparagine, and a phenylalanine. And then for the most favorite one, you can see that this one is positioned quite differently. You have like a glutamate, and you have a asparagine, and then you have a phenylalanine. So this is actually quite interesting because, I mean, I'm going to show like this one here and this one here. They're fairly similar. Glutamate, glutamate, asparagine, asparagine. Here a phenylalanine, and here one a tyrosine, and that makes a huge world difference. I'm not going to talk too much about that because it's unpublished, but it's going to come, I think, a really cool paper about it a little bit later on. Anyway, so since there's either glutamate or glutamine, we chose to look at this. And the work here was done by Kelsey Hall. I'm going to show a picture a little bit later on. Just want to show and to keep up with the, sorry, I have a little bit of a cold, with the title, like History and Future Progress. Marianne, a nice PhD student in our place, she changed the substrate specificity from cellulose to chitin. Sarah and Bastien, they looked at what the importance of these different residues are, and then we also have Mikael Mailetta and his lab at, I don't call this Scripps or Berkeley right now. I mean, it changed place, but he's also been looking into this. So, a little bit off on my animations. I sent it off yesterday, so I cannot change it. But what does this glutamate do? Or glutamine? So Paul Walton and Wang and Carmen Rivera, they suggested by calculations that you have like this caging. So you get hydrogen peroxide, you get a homolytic cleavage, you create an OH radical, which is really reactive. You need to cage it before it reacts with enzyme. Osman, a colleague of mine, and Bastien, they show that also through calculations and also experimental evidence that you have hydrogen bonding in the process of the copper activating hydrogen peroxide, for the hydrogen peroxide pulls off this CH bond here. So confinement, what we call it, is clearly a key. So Lucas, he went down to Austria, he made these mutations. So for this NCA9C, it's a glutamine. We change this to glutamate. We also changed the asparagine and aspartic acid, so we could check on both the functional group and the length of the functional group compared to the copper ion on the substrate. So NCA9C is one of our favorite proteins. It's from Neurospora crassa, chelatic domain connected to the linker. It has a carbohydrate binding module. It's active on cellulose and also active on cellulose oligosaccharides, also active on cellulose oligosaccharides. So it's a very nice enzyme to work with. I'm going to go into detail about that. I think it was first produced by Roland Ludwig's lab, and in our case, we had Kelsey Hall as one of the nicest, best, kindest postdocs I ever had in my lab. Really smart, really kind. Again, this is the reaction scheme. So what did we observe? If you put the mutation in, we see that it acts faster than the wild type. And for LPMOs, all mutations we did originally just kill the enzyme. I mean, so often you do a mutation, you have no activity. Here you get increased activity. So why is that? Considering that these are peroxygenases and we do this just in the presence of O2 and the reductant, we thought that maybe it's because this one here produces more hydrogen peroxide than the wild type. And lo and behold, yeah, comes next. This increased rate comes with a price, because you see, after a certain time, the production of products cease to exist. If we add new ascorbic acid, nothing happens. If you add new ascorbic acid in the enzyme, you get the reaction back going on. So this mutant is faster, but it's also less stable. So we measured the hydrogen production capability, oxidase assay, and we can see that our mutant, the glutamate, is much, much better than the wild type. And that's why you also get a faster reaction. Because here's one more thing. I told Kelsey, can you please plot the redox potential of our LPMOs with their oxidase activity? Because when you want to produce hydrogen peroxide, the first reaction is not thermodynamic favorable. Producing superoxide is anorganic. And then when you put in electrons, you get hydrogen peroxide and so on and so on. It's thermodynamic favorable. So I thought that most likely there's going to be like a correlation between the redox potential and its ability to produce hydrogen peroxide. And then lo and behold, we see a nice correlation. We know that it's not the only factor, but it makes sense that it also is a parameter for this. So basically, it's a better LPMO when it produces its own H2O2. If you provide H2O2, it really sucks. It dies right away. So the wild type is good. Producing, being stable, we put 300 micromolar of hydrogen peroxide, get 300 micromolar of product, and then our mutant, which used to be good, is just really total crap. So again, confirming that if you don't add hydrogen peroxide, you're dependent upon also the LPMO to produce and get the reaction going. If you add hydrogen peroxide, you want things to be, how to put this, chemically correct to contain the reactivity needed or kill your enzyme. I was really happy for this. We're able to publish in JACS for the first time since 2000. We tried to get papers there for like 20 years. They didn't want us, but now all of a sudden, or finally, we got a paper in there again. So, not going to dwell in too much details, but we have this stop flow. We have this other fantastic PhD student, Ole Golten, too unselfish, helps everyone else, produces a lot. And we used to have this postdoc, Ivan Ayuso Fernandez, really smart, spent 3 years with us and moved on back to Spain. Basically, what I'm trying to put here is that we can measure the rate of reduction, we can measure the rate of reoxidation, and we can also measure the rate of electron or radical transfer within the enzyme. I'm going to come back to that a little bit later on. And Ivan was the one setting this up, and Ole was helping. So basically, what we see, there's a huge variance in the rate of reduction. The CVP21 or SMAA10A, the first LPMO we discovered, is really, really fast at being reduced. Our mutant, that is a good producer of hydrogen peroxide, is really slow to be reduced, but if you reoxidize, it's really, really, really fast. And this one is very, very, very slow compared. So that's a huge change. And we use this to sort of like look into the mechanism of how these LPMOs produce hydrogen peroxide. So what is the reason? for this mutant doing this type of chemistry, and we got help from Sergio Giannuzzi and Ashish, and also Serena De Beers did not do the calculation. They're also part of this work from MPI in Mulheim, Germany. And they did some calculations, and I thought it was quite spectacular. That's why I want to show is if you take the wild type, you're going to have one transition state. If you take the mutated to glutamate and you keep it in what you think would be normal in the carboxylate form, because at pH 7 you normally have like COO minus, right? Like acetic acid is always COO minus. At this pH, you also get a transition state and then some intermediates on the way. But if you keep it protonated, the TS is drastically lowered and you go even much further down, and it corresponds well with the reactions we see, both for the reduction and also for the production of hydrogen peroxide. So that's pretty cool. So a consequence is that you have a mechanistic change. Instead of using like this copper oxide, I haven't talked too much about that. That produces the reactive intermediate, and Tomasz has done calculations on the copper oxide and see that it works. You get a change in mechanism. You go to copper 3 hydroxide, form in one single step. And the cool thing here is that the copper oxide has a spin state of plus 1/2. I think. I get confused, keep forgetting. But this copper 3 hydroxide has spin state equals 0. And why is that important? Well, if you don't have spin state 0, like all your electrons paired like this, you're not allowed to react. That's why we don't combust. That's why you can put methane in this room and it doesn't combust. You need to get from here for oxygen, 2 unpaired, to switch either here or use an enzyme to put electrons in, like what you guys are doing typically. So that's what I think is pretty cool. So basically, this one here can react with whatever, and it fits well. And also shows, I think, the importance of computations, and also show the importance of combining computation with experimental evidence. Right, Tomasz? Agree? Thanks. So, yeah, I'm getting close to the end. Like I said, we can also measure how electrons are transferred through the protein, because you can get this tyrosine and tryptophenyl radicals. We didn't show this first. This was shown by Hedison et al. And the guru of copper chemistry, Ed Solomon, at Stanford. Sorry. But what we did do is that we took these sort of like, how to put this? First, to show that for our wild type, after reducing, we can see that we get a tryptophenyl radical increase. We also do the same thing with pyroxyl, and then the decrease. If you have making into asparagine instead of glutamine, we see it exactly the same. But the one that is really inactive, gets inactivated, is really fast at producing hydrogen peroxide. We don't see any transport of electrons. So what we believe is that you get an electron from the center, it goes through this tyrosine, and then we can observe this one and this one here in a thing we call hole hopping. So not only does the second sphere amino acids interact with how they tune the copper center and its electronic properties, it also tunes how you store electrons at the active site. I'm just going to skip this. We took this one step further. Back in 2015, the guru of electron transfer chemistry in proteins, Harry Gray, and his collaborator Jay Winkler, they said that if you have enzymes that are working in an oxidative environment, you're bound to find at least 2 tryptophans and 1 tyrosine in these enzymes. And they're used for what they call hole hopping. So they transfer this oxidative hole that can damage your enzyme through the enzyme and out. And if you look at our favorite enzyme, you have the active site, tryptophan 178, you have tryptophan 119, phenylalanine does not transfer electrons, tryptophan 108, 2 tyrosines, another tyrosine, and a histidine. And it's a lot of people here. George and Julia, they checked that the mutants we made were intact. Roland and Lawrence, they sort of like gave us their technology for measuring hydrogen peroxide consumption. Osman helped Ivan check the stability of the enzyme using advanced Rosetta before producing. And Tom and Ivan, they made all these mutants and made all these different types of reactions. I'm not going to go into detail, but show there's a shitload of experiments. And they checked the electron transfer through the enzyme for all these different types of mutants. And they created quite a few, I think we're like 10 or 15. The main thing is that this tryptophan is really important. If this one goes away, this electron transfer does not take place. The enzyme dies. If you mutate this, you're actually able to increase the rate because you push the electrons back up here. And then if you mutate these here, we observe that the electrons does not get to move out. So this one is the most important to facilitate, and this is the exit out to the solvent. So this we published in Nature Communication this year. So with that, I'm going to summarize. LPMOs are powerful catalysts for oxygen activation and CH hydroxylation. Of course, the histidine brace is vital. We also have this second sphere residues, and I hope that shows that they can affect the copper activity. And also, as postulated by the wizards of electron transfer enzymes, Gray and Winkler, aromatic amino acids are important protection mechanisms in a highly oxidative environment. Yeah. Appreciate meeting. Take questions if you want.